Hello, I'm Tessa Farthing. My research is on the impact of a forested state park buffer on nutrient dynamics in an agriculturally dominated watershed in Southwest Ohio. I work closely with my colleague and co-author Eileen, as well as my advisor, Dr. Grudzinski. So we have these things called non-point source pollutants. Now these are widespread across the landscape, and things like agricultural runoff, and they're very difficult to regulate. As a result of these pollutants, we can have something called eutrophication occur, which can lead to increased algae, anoxic zones, and toxins. Eutrophication is essentially when there are too many nutrients in an aquatic system. Two nutrients of particular concern are phosphorus and nitrogen. In this study, I look at total phosphorus, soluble reactive phosphorus, or orthophosphate, total nitrogen, and nitrate. There are two overarching research questions associated with this study. The primary research question is, how does a large forested state park impact nutrient dynamics in an agriculturally dominated watershed? Secondarily, I'm interested in how discharge, seasonal change, stream geomorphology, and vegetation condition impact nitrogen and phosphorus concentrations. This is the study area of Upper 4 Mile Creek watershed. The boundaries of the watershed fall in 4 counties, 2 of which are in Ohio, Preble and Butler, and 2 of which are in Indiana, Wayne and Union. The central diagram here shows the delineated Huck 12 watersheds that fall within the study area, as well as the central drainage basin of Acton Lake. This diagram over here on the right shows land use and land cover for the study area. This predominant copper color here is cultivated crops. So as you can see, this is an agriculturally dominated watershed. Now there is forested area along the riparian zone of the creeks, as well as this forested state park, which is Houston Woods State Park. The sampling sites are focused around the state park area in Acton Lake. The park boundary is shown here in orange. There are 4 major creeks that drain into Acton Lake. And on each of these 4 creeks, there are 4 sampling sites. One of these is at an inflow point into Acton Lake, another is at the park boundary, and then a third is at a midpoint between these two. The 4th point is located outside of the park boundary on agricultural or residential property. And then this final point is at the outfall point of the lake, where the water is leaving the park. These samples are collected on a bi-weekly basis, and the collection occurs for an entire hydrological year, and that will end this December. After the samples are collected, they're processed in the lab using standard protocols for a Latchet nutrient autoanalyzer. In addition to the water quality parameters, there are a number of supporting independent variables that are collected as well. These include basic morphological features of the creek like the width, the length, and the depth, as well as the creek bed geology and sediment particle size, in addition to hydrological parameters, so how quickly and how much water is traveling through these systems. And then in addition to the lab work and the field work, also use computers quite a bit for remote sensing and GIS to get things like this, which is the NDVI, or the Normal Difference Vegetation Index. And this is a greenness value of pixels in a satellite image, which can be interpreted as vegetation condition. These graphics show the preliminary results of dependent variables. These graphs are for each of the 4 creeks. The mean total phosphorus concentration is shown on the y-axis, and the month that was sampled is shown on the x. And this is for an upstream location, which is at the park boundary point, and a downstream location, which is at infall point into the lake. And as you'll be able to see in all of these graphics I show, for Marshall's Branch and Deer's Ear, generally speaking, the upstream value is greater than the downstream value, but they're following a similar pattern throughout the sampling period. And then for 4 Mile Creek and Little 4 Mile Creek, this pattern is a little less clear, and sometimes the downstream value is actually higher than that upstream value. So this is for phosphorus. So there's total phosphorus here and orthophosphate on this graph. And then this is for nitrogen. So Total nitrogen is shown here, and nitrate is shown in this graph. And again, for Marshall's Branch and Deer's Ear, that upstream location is typically higher than that downstream location. And that pattern is reversed for 4 Mile Creek and Little 4 Mile Creek, where the downstream value is actually equal or higher to the upstream value. But again, seeing a similar change between both throughout the monthly sampling. Moving forward, the rest of the data needs to be collected, including discharge field measurements, pool riffle sequences and pool volume, and those NDVI calculations. After all of the data is collected, analysis has to be carried out in order to answer those two research questions. The first one is to determine that significant difference between the upstream and downstream points. And the next analysis will be a model in order to determine the impact of the independent variables. The model will use water quality parameters as the dependent variable, and the independent variables will be discharge, stream size, land use land cover, sediment particle size, pool volume, and vegetation condition. I want to take a moment to thank all of the facilities, resources, and individuals that made this research possible. And if you have any further questions about the research, you can send me an email at the link provided below. These are my references for my work. And thank you so much for your time.